Yes, good afternoon, class. I welcome you to Metric Hub, our YouTube channel, where education has been simplified anytime, anywhere, real-time education. Please don't forget to connect, subscribe, and keep connected. Like the channel so then you can keep on receiving up to date information with regard to our revision for 2024 examinations. Without wasting any time, please let me just quickly go to the board. I have compiled a number of questions where I'm quite sure that you are going to benefit from them. There is a question on evolution. There is a question on meiosis. There is also a question on probability chance factors of dihybrid crosses. You are also going to find questions on dihybrid crosses. So what I'm saying is these ones that I've just marked here is just some of those, it is just some among others that you are going to find as we do go through the questions. So we have got the diagram that is shown below that shows part of a process involved in the production of protein. I'm sure you remember to say this is called what? Protein synthesis. Before you go anywhere, just go clean and stay, make it that you know. This is protein synthesis. The diagram below represents the production of protein, which is protein synthesis, protein formation. Production can also be saying formation of proteins. Formation of proteins. I'm sure that you must also know that. You must also just take note. Formation of proteins. So that's the diagram that is being shown here. Represents the production or the protein synthesis process, which is shown here. If you go also on our YouTube channel, Metric Hub, if you also follow Metric Hub, you would find out that there is a presentation on what? On protein synthesis. There is also another presentation that is posted already. There is a class on what? On protein synthesis. There is a class on protein synthesis that is posted there, which explains the whole process that is taking place or that takes place during production of proteins. Metric Hub, please don't forget the channel name is called Metric Hub. It's called Metric Hub. It's called Metric Hub. I'm saying on Metric Hub, we also have even business stated, which is also quite synthesized and good and useful for our educational purposes. So let us quickly answer the questions that are there. Identify process Y. Identify molecule Y. The question wants you to identify molecule Y. This is the diagram. Identify molecule Y. What is Y? Like I said, this I have already given this in our notes there in my previous presentation. So please, here I'm just quickly answering the question for the interest of time. So then I can be able to answer all the questions that are attached on the board. But for more information, go to protein synthesis class presentation on the notes which are on the channel. It is quite explained in detail. So here, identify molecule Y. Molecule Y, you can see there we have got that which is a type of RNA that is formed with 3 nitrogen bases, which is complementing or forming against the mRNA. So in other words, I would always want you to understand that mRNA, the single strand, is always mRNA. So therefore, the opposite nitrogen, the opposite RNA molecule that is forming, complementing the nitrogen basis of mRNA, it is called what? Transfer RNA. tRNA, which we can say transfer RNA. So we are saying molecule Y is what? Is transfer RNA. It's called tRNA. Molecule Y, it is called tRNA. And then the group of nitrogen bases, and identify the group of nitrogen bases Z. The group of nitrogen bases Z. Okay, this we are saying on tRNA we have got nitrogen bases. What is the name of these nitrogen bases that are formed or that are found on tRNA? I'm sure that we call them what? Anticodons. A group of 3 nucleotide bases that are found on what? It's called anticodons. Anticodons. It's called anticodons class. These are called anticodons. Nitrogen, 3 nitrogen bases found on tRNA. 3 nitrogen bases found on tRNA. They are called what? They are called anticodons. They are called anticodons. And then the next question is saying if X is the next amino acid required after W, then identify nitrogen bases 1, 2, 3. If X, which is this one, if X is the nitrogen, is the next amino acid required after W, then identify nitrogen bases 1, 2, and 3. What is that? We are saying identify the nitrogen bases 1, 2, and 3. If X is the next amino acid, is the amino acid required after W, then identify nitrogen bases is the next amino acid after W. So then identify the nitrogen bases 1, 2, and 3. So after W, what we have? We have got W there, which is giving us that after W, then identify 1, 2, 3. So 1, 2, 3, we can say 1, it is UGG is going to be UGG. It is 1, 2, 3. It's going to give us uracil, guanine. If X is the amino acid that is required after W, identify that. We are saying 1, 2, 3. It must be uracil, guanine. 1, 2, 3. It is supposed to be uracil, guanine, which is there. And then we are saying identify the nitrogen base, identify the DNA base triplet that codes for X. The DNA base triplet that code for X. What is the DNA base triplet that code for X? So we are saying this if X is equals to X is given as UGG. Therefore, this is what? This is tRNA. So the question is saying identify the DNA base triplet that code for X. So obviously, if it is a DNA, therefore we should have what? we should on T we should have G. I mean we should. I mean on U we should have T, and then obviously we have got what the guanine. So in this case, what you need to understand is that it's just for you to know that this U, it is found on what, on RNA, and then if it is if it is DNA, it has got a what? It has got a thymine. So the question is only the question wants you just to understand that we have got uracil in what, in RNA, and we have got thymine in what, in DNA. This is in what, in DNA base triplet of UGG. In terms of DNA, it is supposed to be what? It is supposed to be TGG. It is supposed to be TGG, which is suiting in the correct order according to UGG that we have found there, as to say this is for what? This is for RNA, and then in terms for DNA, it must be in this case. Please, class, don't make mistakes. Take note of this. And then describe the process of transcription. Transcription, basically, we are talking about the process through which RNA is formed. Process by which RNA is formed is what we call transcription. Is what we call transcription. So the question wants you to describe the process. Describe the process of transcription. We can say the description, the double helix DNA unwinds DNA. Double helix, it must unwinds. It unwinds and unzip. It unwinds and unzip, causing also the weak hydrogen bonds to break. The weak hydrogen bonds. The weak hydrogen bonds to break. Forming two strands. When the weak hydrogen bonds break, the forming two separate strands of DNA, forming two separate strands of DNA. That is what happens there. In that regard, one strand of DNA is used as a template. One strand is used as a template to form mRNA. To form mRNA. I'm sure that messenger mRNA, you know, that is messenger mRNA, it is formed because of one template, one strand of DNA that is used as a template. And then next thing is that we have got using the free floating nucleotides. Using the free floating nucleotides from the nucleoplasm. From the nucleoplasm, we are saying the mRNA is the mRNA is complementarily pairs to DNA. It complementarily pairs to the DNA strand. To the DNA strand, what happens here is that we have got a small piece of DNA that unwinds, and then from the free-floating nucleotide bases which are in the nucleoplasm, in mRNA, it complementarily pairs to DNA. In this case, what is supposed to be understood is that the coded message for protein synthesis is thus copied to mRNA. In that case, the coded message for protein formation, which is from DNA, the coded message for protein formation contained in the DNA is thus copied to mRNA. Is therefore copied to mRNA. And then the small piece, the small single strand, of mRNA breaks off and lay off the cell of it breaks off and lie down alone. So what we are saying is that please understand that the process of DNA, the process of mRNA formation is what we call transcription. The process of transcription is the formation of mRNA. So we are saying it is formed as a result of the small piece of DNA unwinding and breaking off the weak hydrogen bonds, such that the two strands of DNA are used as a template, where one strand basically is going to be used to form complementary bases or to form complementary bases that pairs up to it using the free-floating nucleotide bases which are found in the nuclear plasm. And then the coded message for protein formation is therefore copied to mRNA. What you need to understand is that the message must be encoded to mRNA such that we are saying that is transcription. Therefore, that small piece of that small single strand, the single strand of the mRNA breaks off and lie down alone, just as like that. Therefore, we can explain that the process of transcription has taken place. We have got the next question. In certain marine invertebrates, the color of the shell is under the control of one gene with 3 alleles. Is under the control of one gene with 3 alleles. In different combinations, the 3 alleles produce 4 phenotypes. The 4 phenotypes, we are saying the outward appearances. Those are the 4 phenotypes. We are saying we have got what? We have got the 4 phenotypes, which we can say we have got 1 orange, we have got 2, which is yellow, we have got 3 orange yellow, and then we have got 4, which is black. So the 4 phenotypes are given there. The table below shows the results of the offsprings produced from crosses involving parents of different phenotypes, which we are saying that is what mating, from mating parents of different phenotypes. We have got the phenotypes of shells that are given there. Then we are saying, the question says, we have got cross 1 is yellow, yellow cross together, it gives us 27 yellow and 9 black. And then we have got black, it gives us all black. And then we have got orange and orange, it gives us 30 orange and 10 black. We have got orange yellow as number 4, and then it gives us all orange. So the question is asking us to, here we are supposed to understand the processes or the terms of recessive and dominant alleles that are found in organisms. To say a dominant allele is the one that would always appear in the phenotype. Hence, the recessive allele can never be visible. We cannot see it in the phenotype of an organism. So that is one key thing that you must understand with regard to this question. So the question is saying, name and describe the type of dominance shown by cross 4. Cross 4, we are saying we have got orange and yellow. Cross 4, we crossed cross 4. Orange was crossed with what? With yellow. And then we have got all yellow, orange. We have got all showing all the offspring showed or produced a blend, or they showed both of the phenotypes that were used in the cross. So the question is saying name and describe the process. So naming, first thing is what it is. When we have both of them showing equally the same, we have got what is called codominance. It is called codominance. It is called codominance. The type of dominance, please understand that codominance is when both of the phenotypes are shown in the offspring that are being produced, and then nothing of them is dominant over the other. And then again, in terms of complete dominance, it means one color can dominate the other, where we have got two different phenotypes crossed, but we have got one dominating over the other, which is that we call that complete dominance. So the codominance is that the phenotypes of or the alleles of the parents are equally expressed. Alleles of the parents are equally expressed. That is, equally expressed. That is codominant. Are equally expressed. That is codominant. And then both are expressed and both are visible. Both are visible on the phenotypes of the offspring. On the phenotypes of offsprings. So that is codominance. That is codominance. That is codominant. And then the next question is saying, which shell color is controlled by the recessive allele? Which shell color is controlled by the recessive allele? We are saying which one is controlled by the recessive allele. We can check it by checking here to say that, for example, we know that recessive allele can only be expressed when it is crossed against itself. So we are saying in this case where we have got yellow orange, yellow, we have got others yellow and others black. So in this case we have got what you call cross number 2. Cross number 2, we have got black and all black. So in that case we can basically say that it can be controlled by a recessive allele, which is what? Which is black. Which is black in terms of question number 1, 3.2.1. 3.2, 2.3.2. It is black. And then use the information in the table to support your answer to question above. So we are saying we can use that to support that one. Use the information in this case to support the scenario. We can say in cross 1, both parents are yellow, and none of them are what? Are black. We are saying in cross 1, in regard to this question, in cross one, which I've already showed you there, in cross one, both parents are yellow. In cross one, both the parents are yellow. As we can see, none of the but black appears in the phenotypes of the offsprings. But black appears in the offsprings. That is quite true. We are saying they are both yellow here. And then what is happening here, we can even prove it mathematically, which we can say that in this cross number one, we can say we have got a homozygous yellow and a heterozygous yellow. In this case, we have got, we can say that it can be what? It can be homozygous yellow and heterozygous yellow, which I can basically say that I can show you this as to say, what are we, to show you what we are saying. Cross one, all were yellow, but we have got 27 yellow, and then we have got 9 black. This can be proven by saying, we can say this can be shown by having a yellow, homozygous is crossed with what? With a yellow heterozygous, which can basically be the same, can give us a chance of what? Of saying there is a black in it. That can be one case. And then in cross 3 again, if we look in cross 3, again, cross 3, which is given here, cross 3, we say, oh, they were orange, and then cross 3, we have got all orange crossed, and then they give us what? 30 orange and 10 black. Hence, it means black is inside the orange parents, and then it is hidden there. So we can say in cross 3, both parents is another alternative that we can give there in regard to this. In cross 3, both parents were yellow, were orange, sorry. Both parents were orange, but what happened? But black appears in the phenotypes of the offsprings. Black appears, but black appears in the offsprings then. So hence we can also be able to conclude to say that there is what? Black is the recessive allele because it is hidden in the parents. It is a recessive allele that is masked by yellow, it is masked by orange, which we are saying at the end of the day, it is appearing in the blood as what? As being hidden, and then it comes out as in what? In the offsprings. It also indicates that those were also a kind of a what? Of a recessive allele. I'm just trying to calculate again on cross 1 that can also be shown. Remember to say in complete dominance, I'm sure that you remember that in cross 1, just want to illustrate something that I'm picking up again here, that I want to show you here. In cross 1 again, in cross 1, I want you to see something. In cross 1, we have got, in cross 1, we have got yellow, and yellow crossed together. So what is happening there? If we can look on the if we can look at this, the outward or the final answers, the offsprings that we have got a ratio, or our answer that we get, they give us 27 s to 1, I mean s to 9, sorry. The answers that we gave on the offspring is 27 s to 9, which we can typically prove by saying this can be that can be what to say. We have got yellow, it can be just yellow, which can give us to say that we can have a ratio of what? Of saying this yellow recessive and that yellow recessive there. It gives us an F2 generation, an F2 generation of what? Of monohybrid cross, of monohybrid cross, which we can find out that in F2 generation, what does it do? It always gives us a ratio of 3s to 1. It gives us a ratio of 3s to 1, which we say that 3 is the dominant allele, 3 dominant s to 3 dominant s to 1 recessive, which we can say that in this case our yellow is dominant s to black, that is also recessive, s to black, that is recessive there. 3 is dominant s to black, that is recessive. I'm just mathematically working it out from our understanding, which you remember that the monohybrid cross, it gives us a what? An F2 generation of 3s to 1, which is given by this ratio of 27 s to what? S to 9. We can simplify or reduce it to say that can give us, it can give us what? 3 s to 1, 9 s to 27 s to 3 s to 9 can give us 3 s to 1, which is quite correct and very true to say that black is what? Black is recessive. Black is recessive. Yellow is dominant, or either even orange is also dominant, and then black is recessive. I'm sure that it is quite interesting and making you to understand exactly what we are talking about in this regard. Thank you again, and then let's move on for the interest of time. Let's quickly move on. The back of a leopard frog, uh, which is runner pippins, the back of the leopard frog, which can be spotted as shown below or without spots. The back, it can be spotted, which is as shown in this case. And then we have got spotted frogs were allowed to interbreed, and they produce 150 spotted offsprings. They produce 150 spotted offsprings, and I mean offspring, and 50 offspring without spots. So we get 150 s to 50, 150 that are spotted s to 50, which are what? Not spotted, s to 50 not spotted. That's what we are having when the breeding took place. And then the question is saying which phenotype is dominant, in that regard. Which phenotype is dominant? Obviously, as you can see, we are saying the dominant phenotype, we are saying the spotted and non-spotted. The spotted frogs were allowed to interbreed with as they produced that one. So in this case, we are saying 150 are spotted and 50 are not. So again, it is referred, if we refer it again to the relativeness of the previous question, we can see that the one which is dominant would always have the highest ratio. So we are saying spotted back is what? Is dominant. Spotted back is dominant. spotted back is dominant. Explain your answer in that regard. We are saying spotted frogs produce, we are saying spotted frogs produce offsprings without spots. The spotted frogs produce the frog. I mean, this is similar to our previous answer that I just answered above there. So we are saying the spotted frogs produce, I mean, spotted frogs produce offsprings without spots. Spotted frogs produce offsprings which were without spots. So the without spots was just hidden inside the what? The spotted. So what we are saying is that this was a spotted frog, but it also have a recessive gene of what? Of not having spots. So the dominant would always be able to what? To master and to, I mean, to dominate. So the spotted frogs produced the ratio, I mean, it produced the thing, the highest, which we can as well again, if we have got this ratio here explaining this answer, we are saying we have got 150 spotted, which is as to 50 not non-spotted. This can as well be traced back to further if we further simplify it, it gives us a ratio of saying we get also a ratio of 3 as to 1, which we can simplify by dividing by 50. We divide by 50 there. We are saying 150, because for the interest of others, just to make things clear, how do we get this? We are dividing by 50. We are just simplifying our maths here. We divide by 50, we divide by 50 here. If we divide by 50 there, if we divide by 50, we are saying 50 into 50 there, we get 1. 50 into 150 here, it comes in 3 times. That is where we are getting the ratio of 3 as to 1. That is where we are getting 3 as to 1. So 3 as to 1, the dominant is always given by what? By having our dominant allele is always the one that has got a ratio of what? The bigger ratio, which is similar to the previous question that we just worked there. So 3 as to 1, 3 spotted and 1 are not spotted. The spotted one is the dominant allele there. A frog that is heterozygous for spotted back was crossed with a frog without spots. A spotted frog was crossed with, I mean, a spotted frog, I mean, a frog that is heterozygous for spotted back was crossed with a frog without spots using letters D and recessive D represent a genetic cross to show the expected phenotypes and genotypes and phenotypes of the F1 generation. Class, this one I always say that you must master and make sure that you get all the marks. This we can do it this way, just to for the interest of time. We have got our P1, and then we have got our phenotype. We have our phenotype. Our phenotype, we are saying what is happening here. We are saying spotted frog, spotted, it is crossed with no spots, is crossed with no spots. So the question is saying it is a heterozygous. The question is saying we have got a heterozygous here, heterozygous spotted. So we are told to use the letters D there. So we must understand that the genotype, it is supposed to be D. D is crossed with what? With recessive D. And then in that case, we must understand that meiosis must take place. Meiosis must occur, and then we have our gametes, which we are going to say our gametes, we have got this D, we have got that D here, and then it's crossed with that single D here, because as you can see there, it is what? It is meiosis, it divides by 2, therefore we can have one recessive D there. And then we say fertilization, which is the fusion of the gametes. So we have got this D here, it comes this D, and then we have got that D there, it goes there. And then we have got this D, and then it comes with this other recessive D. So we can have also that D and that D, then we can have this. So this is our genotype. This is our genotype, and then we are saying that is our genotype, and then we say phenotype. Phenotype, we are saying one spotted as to one not spotted. So the question is saying what? The question wants us to say use the represent the genetic cross, show the expected genotypes and phenotypes. So we are just asked to work it until up to here. It's one spotted as to what? As to one non-spotted, which is going to give us our answer as correct as it is. Maybe others, they are used to work this question with a Punnett square. Let me just quickly just illustrate it to make it clear to others, so therefore they cannot make mistakes here. What is happening here is that we can have our gametes there with a Punnett square. I'm just trying to make sure that you can as well be able to what? To understand what we are saying. Through a Punnett square. So we have got our spotted. Our spotted, we said it is DD. It is DD there. Let me just quickly do something here, adjust it, pull it this side. Our spotted, we have got DD there. Just to quickly work it out, we have got G and G there, and then we have got not spotted. Not spotted is now down here. We have got this D and that D. We just take this is not spotted. So we take it down, and then this is the spotted, we take it there. So it is this D, we put it there, is this D, we put it there, and then it is this D, and we take it there, is this D, we take it there. We are just saying it is the fusion. And then we have got this D and that D, we have got this D, it comes down there, and the small d, it goes there. We have got this d here, which is the small d, and that small d there, we have it like that. So when we go through a Punnett square, it will take us to say we have got two spotted and two spotted and two what? Not spotted. So it will give us a ratio of saying we have got two spotted as to two not spotted. So this can just be further simplified by dividing by 2. You divide by 2, and then you get one spotted as to one not spotted. Then you get your answers correct there. You see. So I'm saying, please take note that this is just something easy and quick. Take all your 6 marks and don't ever turn back. Go and grab them and get the best there. Just the last question for today. It is just our last question of the day, which we just want to quickly answer there. It is just diagram below shows the skulls of two species of primates. There are two species of primates, skull 1 and skull 2. Tabulate the observable differences between skull 1 and skull 2 that show trends in human evolution. Tabulate the observable differences in these skulls, skull 1 and skull 2. So if you are saying, when the question says tabulate, it means it wants you to give them in a table form. It wants you to give this in a table form. It wants you to give this in a table form. So what we are saying is that we can draw the table there. We can draw our table and then we can say skull 1, skull 1, and then there we divide it. And then we can say skull 2. So what we want the differences that are between them. We can say 1, they are quite, this one, skull 1, it has got a sloping face, as you can see. It has got a sloping face here. Skull 2, as you can see, it has got something like a flat face. That is 1. 2, 2 there, the observable difference is there. We have got skull 1, it has got a poorly developed cheek or chin. Poorly developed. It has got a poorly developed chin, and then this one has got a well-developed chin. Well-developed chin in skull 2. Number 3, we are saying the chin here, this is number 2. Number 2. Number 3, we can say this one, skull number 1, it has got large jaws. As you can see there are large jaws here. It has got large jaws, and then this one, as you can see, it has got smaller jaws. It has got smaller jaws. Skull number 2. Question number 4. Another difference that we can give there, that I can give again, this one, it has got a small cranium size. As you can see, the cranium size versus this one, we are saying number 4, it has got skull number 1 has got small cranium size. As you can see, this one has got what? It's got larger cranium size. Larger cranium. I'm saying if you can look here, skull number 2, it has got larger cranium size. It has got small cranium size, which is some observable differences that you can see there. I think I can as well say that it is one number maybe one other difference that I can give again. It has got more protruding jaws, number 1. More protruding jaws, which we can say that it is what? It is prognathous. Number 5, it is prognathous, this one. And then number 2, it is actually non-progenitus, this one. It is non-progenitus. It is non-progenitus. Can you see those differences? Please just take note. We are saying when you are tabulating, don't make a mistake of doing anything else. When the question says tabulate, it wants you to draw a table which we can comparatively analyze or assess the variables or the information that you are given. So that's what you need to do here. So it is like that. Then you get your 7 marks, which is 1 for the table, and then 3 times each. Here I just gave you as many as possible. Since it is just a revision, I just gave you as many as I can, but it needed at least you have 3 of them. The next question wants you to say, it wants you to answer, give 4 characteristics of the upper limbs that humans share with other primates. Give 4 of these. We are saying give 4. This question here, question 3, we say 1, give 4 of them. They've got freely rotating arms. The arms are freely rotating. 2, we can also say they can also have opposable thumbs. I'm sure that you know opposable thumbs. And then we can also talk about 5 fingers. This is number 3. Other primates have got also 5 fingers. Number 4, we can also talk about they also have bare fingertips, or they have got nails instead of claws. Instead of claws, they have got nails instead of claws. And then number 5, what else can we give there? They've got longer upper arms. Longer upper arms can also be there. One other point that I still I can as well add to this is that they also have fingerprints present. We also talk about fingerprints present. I think that can as well be quite good to add that. And then we can just give any 4 of them. We gave 6 of them. Number 3.14, explain how an increase in cranial volume is related to intelligence. The cranium actually is the one that houses the brain. So one, we are saying since the cranium houses the brain, since the cranium houses the brain, a larger cranium volume indicates larger brain. It indicates larger brains. indicates larger brains, which would also suggest greater intelligence. Suggesting greater intelligence. So in other words, we are saying the bigger the cranium, the larger the brain, which would also help to suggest greater reasoning. Again, the larger the brain or the bigger the brain, the better the reasoning as well. Next question is saying humans are bipedal organisms. What is meant by bipedalism? Bipedalism is just meaning to say walking on two legs. Walking on two legs. Walking on two legs is bipedalism. And then explain how each of the following skeletal structures have contributed to bipedalism in humans. One, the foramen magnum. This one, it moved to a more forward position. It moved to a more forward position. It moved to a more forward position to allow the spinal cord to enter the vertical. To allow the spinal cord to enter vertically. And then the pelvic is actually short. It is short and wide to support the upper body. Is short and wide to support the upper body. And then the spine is actually is more curved. The spine is more curved or S-shaped. It is more S-shaped to absorb shock. To absorb shock. To absorb shock and also allow flexible movement. Flexible movement. These are the things there. I'm saying, how do these allow bipedalism, the foramen magnum, the pelvic, gentle, the spine? So basically, please take note of these. You need to know that these are quite well explained. And then geographic isolation, it is just for the interest of time. Check it again. It has been explained with more diagrams in previous presentations. It is well explained with more diagrams in previous presentations that are on our channel. Just refer. There is geographic isolation or geographic speciation, which is well explained there. Let us just finish the last part of our question, which is mutation in gene allows terpenes. It allows terpenes it allows terpenes to survive at high altitude. It is possible to cope with the low oxygen content at high altitude there. One way is for the body to produce more red cells in response to an increase in altitude. Another way of coping has developed in terpenes as a result of a gene mutation that they inherited from their ancestors. The mutant gene helps them to use the low amount of oxygen present more efficiently. The mutant gene was found in 87% of the terpentine population, but only 9% of the honey population that live at low altitude than the terpentines. So the question is saying 3.4.1, what does it want us to do? A gene mutation caused variation between the terpentine population and the honey population, named through other sources of variation in human population. A source of variation, we are saying in human population, what causes variation? We have got one, crossing over. Crossing over, it causes variation. We have got random mating as another point there. Number 2, random mating. We have got number 3. Number 3, we can also talk about random fertilization. Random fertilization, when fertilization is taking place, millions of sperm cells are produced, but the fattest and the fastest one can fertilize. And then we can also talk about random arrangement of chromosomes. Random arrangement of chromosomes. Random arrangement of chromosomes. So please class note that all these we talk about crossing over and random assortment of chromosomes. We just refer them as to say they are causes of variation because of meiosis, which is crossing over, and also we can also say what the random arrangement. And the random arrangement of chromosomes. So please class, take note of this. Question number 3.42. Give evidence in the extract which suggests that the survival of people living at altitude could be due to genetically inherited trait. We are saying from the extract. So we are saying 3.4.2, we are saying give the evidence in the extract which suggests that the survival of people living at altitude could be due to genetically inherited trait. So that is A. So you can see that they inherited, we are saying another way of copying this is in the table that is as a result of gene mutation that they inherited from their ancestors. The mutant gene, we are saying inherited from their ancestors, and then also it's saying the mutant gene also there. So we are saying it can be this, it can be also because of this, the inheritance. So the answer is that due to genetically inherited trait. If it is genetically inherited, we are saying therefore it can be what? It can be one, we are saying it is a mutant, or else we also have to say or it's inherited from the ancestors, as to say or inherited. Inherited from what? From ancestors. inherited from ancestors. That can also be quite good. And then give one evidence that suggests that the survival of people living at high altitude could be caused by environmental factor. Can be caused by environmental factor. We are saying, how can it be caused by environmental factor? This one way of red blood cells, a gene mutant, let me just help from them to use low amount of oxygen. They also, we are saying low environmental factor, we are saying this can also be low amount to use low amount of oxygen. Use low amount of oxygen. The mutant gene help them to use the low amount of oxygen or less oxygen, and then that is at a lower altitude than that. The terpene has got a gene mutant found in 85% of that. So what is that we are saying? It is influenced by what? It caused by environmental factors. It is influenced by what? By altitude. Altitude is affecting, is influenced by altitude. It is influenced by altitude, that we can say. It is environmental, caused by environmental factor. It is influenced by what? By altitude. And then number 3.4 point here, 3.4.3, explain the advantage of producing more red blood cells. Advantage of producing more red blood cells. And then I'm sure that you can understand that the function here, you just need to understand the function of the red blood cells. So we are saying one, more homoglobin present, more homoglobin is present. More homoglobin is present. And then what else? To allow maximum absorption of what? To allow maximum absorption of what? Of oxygen. To allow maximum absorption more absorption of oxygen, which is needed for their needs. More homoglobin is present to allow maximum absorption of oxygen needed for what? To meet their energy needs. More absorption of oxygen to meet energy needs. To meet energy needs. Let's just finish the last one. Hey, the last part, describe how Lamarck would have explained the survival of turbines at high altitude. It's a long story here. Let me just quickly paraphrase it. You know the Lamarck's theory, which talks of the theory of Lamarck. What are we supposed to say? One, originally the amount of red blood cells was similar in all humans. Originally, the amount of red blood cells was similar in all humans. Was similar in all humans as a result of low oxygen content at high altitude. As a result of low oxygen, as a result of low oxygen content At high altitude, the red blood cells try to increase the amount of oxygen absorbed. The red blood cells tried to increase the amount of oxygen absorbed. The amount of oxygen absorbed, try to increase the amount of oxygen absorbed. That is it. And then we are saying, as a result, ancestral terpenes produce more red blood cells. As a result, ancestral terpenes produce more red blood cells. Produce more red blood cells to increase the availability of oxygen to the body. To increase the amount of oxygen to the body. The amount of oxygen to the body. This acquired characteristic, was passed on to their offsprings. Was passed on to their offsprings. Was passed on to the offsprings. And then we are saying all terpenes now produce more red blood cells. They produce more red blood cells. They produce more red blood cells to survive at high altitude. So basically, this is the story, guys. Classic. I want you to understand that Lamarck's theory, you identify the characteristic that is not there, which therefore developed. So at first, start to say Lamarck nullified the idea of variation. So all organisms were born the same. But now we are saying there is a deterministic theory or a deterministic or acquired characteristic that you must pick up here. So from this scenario, we are saying there is an issue of saying the amount of red blood cells was similar in all humans as a result of low oxygen content at high altitude, that the red blood cells try to increase the amount of oxygen absorbed, and therefore as a result ancestral terpenes produce more red blood cells to increase the amount of oxygen that the body required. This acquired characteristic, and therefore we are saying this acquired characteristic was passed on to their offsprings, and therefore all terpenes now produce red blood cells to survive at high altitude. So that's how Lamarck's theory could have been explained, or that's how Lama could have explained the survival of terpentines at high altitude there. So thank you very much for your time, and please don't forget to follow and subscribe the channel and share it to friends, so then we can all pass it. Let's all go to varsity, let's all go to some high level programs there. Thank you very much and have a beautiful day. I thank you.